hopefully you'll be able to read the slides from here on. Welcome everybody again to the second session. I'd like to introduce our first speaker, Dr. Alessandro Massolo. He's a professor at the Faculty of Veterinary Medicine at the University of Calgary, and he'll be speaking to us about Echinococcus multilocularis in Western Canada.
Hello everybody, and thanks for inviting me here. Thanks to Melissa. And yes, I will be talking about, I don't know how many of you are familiar with this little guy here. I want to do this. I like doing this. Is that okay for you? I need to walk. I'm Italian. I'm over-talkative. So 20 minutes is really not enough. And 15, oh, it's a drama. And we are very drama, right? Okay, so this is the guy we'll be talking about today. It's a very small tapeworm that lives in the intestinal tract of definitive hosts like wild and domestic canids. So foxes, coyotes in North America, and dogs, and eventually wolves. The adult that is in the intestinal tract shed eggs in the feces of the definitive host, and these feces contaminate the environment. When they are accidentally ingested by the intermediate host, most of the time is a rodent, they hatch in the stomach of the rodent, and an oncosphere is released. It reaches the intestinal lumen and passes through the intestinal wall, reaches the blood vessels, and gets to the target organ, most of the time the liver. The problem is that when they are in the liver, they develop into a metacestode stage that starts an asexual multiplication over and over and over, getting from a few hundreds to hundreds of thousands of larvae. After a certain amount of time, that is very often depending on the species of host, they develop in the final stage, the infectious stage, that is the protoscolex. And so in this stage here, when you have hundreds of thousands of those protoscolices, if the rodent is preyed upon by a competent definitive host, then the protoscolex will migrate from the stomach of the definitive host into its intestinal tract, and with the hooks will attach the intestinal wall and develop into the adult worm. And the consequences for the rodents are really dramatic, to an extent that we believe there is some kind of parasitic manipulation of the ability of the intermediate host to escape predation. So the parasite is likely to affect the likelihood of an infectious rodents to be preyed by a competent host. Occasionally, if these eggs that are in the environment are accidentally ingested by other hosts, they may develop diseases or infection. I call these aberrant or accidental hosts. What happens in the intermediate host, so in the rodent? So as I mentioned to you, there is this asexual multiplication of the metacestode larva, and when that happens, it's like a cancer disease. So there is an infiltrative kind of damage, and there are these lesions. This is very different from what we are used to, that is the cystic echinococcus, that is very localized, and the cyst is growing, and all the multiplication is inside the cyst. In this case, there is a multifocal kind of infection, and so that's why this disease is called alveolar echinococcus, and not cystic echinococcus. So this is pretty different from the infection caused by the Echinococcus granulosus complex species, okay? Another thing that is different between the two species is that the fact that granulosus complex species are circulating between, most of the time, these canid species, so foxes, coyotes, wolves, jackals, you name it, even lions in Africa, and ungulates. So in North America, the ungulates are the intermediate host. So it's a completely different cycle. The two worms are very similar. The granulosus complex, that in North America is called Echinococcus canadensis, so the species in North America. It's a little bit bigger, but you cannot tell them apart morphologically, so you have to do molecular identification. So this is the kind of lesions that when these vesicles, when these lesions erupt out of the liver, the asexual multiplication continues in the body cavity and becomes like a metastatic disease. It can be very severe. Infection can reach other organs like brain, lungs, and in some cases, dogs that are definitive hosts, so they are supposed to be immune to the infection by eggs. They should be competent only for infection by protoscolosis. In some cases, only dogs, not coyotes, not foxes, never reported for these wild species, but in dogs, occasionally they develop the alveolar echinococcus disease. So the eggs that they probably ingest accidentally by eating other dogs' feces, or simply because they are shedding. They are an intestinal infection with adult worms, and they are shedding eggs, and they clean themselves and they ingest the eggs. So normally these eggs are not infectious, but in some cases they are. And these dogs, usually young dogs or immunodepressed dogs, they develop a very quick and severe disease, and they die in a few months. Unfortunately, what is happening is that in some cases, in some conditions, even people can develop the disease. And as you can see here, the kind of lesions in the liver at an advanced stage are fatal. So people die of it, okay? So there's some statistics that are reported is that the difficult thing is that this is a chronic disease that can develop after 5-10 years after the infection. So it's very, very difficult to track down. It's much less, it's much faster, the development of disease in immunocompromised patients that usually are considered sentinels. And we are talking about people here, okay? And if it is not treated, is a deadly disease in 95% of the cases. But unfortunately, the mortality even in treated patients is pretty high, 12%. And even if you are treated, your expectancy of life is reduced, so 19 years is estimated. So usually the key transmission routes for infection to people are two. One has been, let's say, the one that is more convincing is the one through vegetables and berries. For example, in Europe, you know that when you pick up your berries and strawberries or blueberries, you want to wash them very well, very much. I always go in the mountains. I just eat them, but it's okay. But the recommendation, they say, wash them. So what we have done a few years ago when I arrived at the Faculty of Veterinary Medicine, I didn't know what to do, to be honest. They offered me a job. I was from Italy. There was no job in Italy. I was told, okay, now you do what you want. What do you mean you do what you want? So I said, okay, let's study. I love dogs. I was studying wolves. So I said, okay, maybe I can study wolves. But then, no, I don't have money. Wolves are difficult to study. I want a good model species. So I started working on coyotes, okay? They are perfect model species, and they're marvelous animals. They're very clever. So in the city, we started working with the city of Calgary, and we said, okay, what is happening to the coyote population? So the first thing that was easy to do is just to collect carcasses of coyotes that were found in the city of Calgary. Do you have an idea how many coyotes are picked up? So the one on the roads in the city of Calgary every year. Any guess? 100, 150 every year. Over a population that we have estimated to be about 800, 900 over the whole city. Okay, it's a completely different landscape from Vancouver, as probably many of you know. So what we did was we collected all of these, and we washed the intestinal tract to detect the adults. And so what we found that with the very, very rough at the beginning, we were just starting, so we didn't really know what we were doing. And I was an ecologist. I was not a wildlife health ecologist, although I was hired as a wildlife health ecologist. So I didn't really know what I was doing. But even with that poor background, we detected 1 positive coyote every 4, so 25% in the city of Calgary. But then when we started considering that this was relevant, we started developing better techniques, and this estimate went up to 40, 45, 50%. So the first estimate was based on only 91 coyotes, but then we have collected more than 300 coyotes in these years. And the estimate of prevalence has stabilized around 45% in the city. 72% in mangy coyotes, okay? So when you think about 800, 900 coyotes in the city of Calgary, those estimates give you the idea that about 200 or 300 coyotes are shedding Echinococcus multilocularis in the city at any time. So this was the, we also investigated which were the main intermediate hosts, and the most relevant is probably the meadow voles, and the deer mouse. Those are the key players in this transmission route. Given that in Calgary we have a lot of off-leash areas where dogs and people walk together, and of course they interact with coyotes. And one of my former PhD students, Anya Smith, is here. She collected more than 900 fecal samples of dogs. No, more than that probably. And so she did a huge study on Giardia cryptosporidium, but we are not talking about these now. But certainly the problem is that when you have such an overlap between wildlife and domestic animals, of course you wonder, is there any risk of transmission? But when I started talking about that, I was asked, so, but what is the situation in humans? So what is actually the situation across the world and in North America? Oh, sorry, before I go there, I wanted to tell you that in 220 samples that Anya collected for her work, we actually looked into them for assessing the prevalence of Echinococcus in dogs. And what we found was 2 positive cases. And I will talk about the rest later on. But even if you think you consider this to be a high estimate, so 1% would be a high estimate. Even if you consider a much lower prevalence, a much lower prevalence of 0.1, 0.5%, we are talking about from 100 to 600 positive dogs that are shedding in houses in the city of Calgary. Interestingly, when we started looking about the liver cases of infection, so alveolar echinococcus in dogs, when we actually started looking into it, we found cases. So we just started 1.5 years ago, and we already found 8 cases of alveolar echinococcus in dogs. And this is creating a lot of troubles for practitioners, vet practitioners, because they don't really know how to treat that. So what is the situation in humans? So every year across the world, there are more than 18,000 new cases, okay? 95% of them are in China, in the Tibetan Plateau. And 150 now, the number is increasing, so about 200 cases, new cases, every year in Europe. What about North America? Well, we know that there were a few cases, some tens of years ago, so in the 50s and 60s, there were some cases in St. Lawrence Island in Alaska, due probably to some movement of a parasite from China. And only 1 case in Minnesota was reported in the 70s, and 1 case in the late 20s in Manitoba. So the overall idea was probably the North American Echinococcus is not that dangerous because we don't have cases. And that, to be honest, was not really a scientific kind of evidence, but it was reasonable. But guess what? After we started talking about that, I started talking to human health people, and 9 months after I went to the University of Alberta to give a speech like this, guess what? A new case, the 3rd case in North America was discovered in Edmonton. There are many more cases, just to make sure we understand, I explain myself very clearly. There are many more cases in North America, but most of them were travelers. So people that were exposed somewhere else. So the cases that I mentioned to you so far are cases acquired locally, so in North America. So this lady was an immunosuppressed patient, 49 years old. She had a kidney transplant. She was negative to serology before and positive to serology after the transplant. And she was diagnosed in May, and we confirmed with qPCR, we confirmed the infection. Actually, we couldn't use qPCR. We had to use droplet digital PCR because the qPCR was not sensitive enough with the bioptic samples that we had. Interestingly, she lives on a farm southwest of Edmonton. She has dogs, 3 dogs, and she normally produces her own vegetables in an open garden, open to coyotes, foxes, and dogs. The one thing that is important to clarify is that across the world, this parasite exists in different strains. Let's say 3 main strains. So one strain is the one in Asia, okay? One strain and you can see that in Asian was both here and here. One strain is red in Europe. And then there was a North American tundra zone strain and Central Europe. The point is that some people starting looking into the kind of strain we had in North America. So Emily Jenkins from the University of Saskatchewan, working with Peregrine in Guelph. And so, 2 minutes, oh my God. For the 15, right? Can I take some question time? I told you I'm talkative. So we decided to look into rural and urban. And we collected more than 270 coyotes. We do the usual work, so intestinal washing and qPCR. And then we strain type. And what we found that almost all the animals, all the specimens of Echinococcus that we found in dogs, coyotes, and rodents were European-like and not North American strain. When we looked into Edmonton, 7 out of 8 coyotes were carrying European-like strain. When we look into rural area, all of them were European-like. I say European-like because we don't really know if this is an undiscovered strain or existing already, or it was actually an invasion. So what we did was doing microsatellite analysis on the EMSB target. Now this is a little bit complicated, but the point being is that all the elements that cluster together belong to the same haplotype, to the same genotype. And when you have a very low cluster like this, it means that these are very, very similar to each other. They have a very low variability. So what we did was we pulled together our samples with this technique, with the samples from sample worldwide. So these are the samples from North America, Alaska, and Europe. And this is an expansion area in Europe, and this is the traditional core endemic area in Europe. And of course, we had a case of North America in 1 fox and not coyote. All our rural and urban Alberta fell very close to the core endemic type. And I would like to show you, to emphasize this low variability in our samples. And the Saskatchewan ones, you see, very, very low variability, and again, close more to the expansion area. So European-like. So what we are working on is understanding whether this low genetic variability is an indication of a potential invasion, recent invasion. Through what? My working hypothesis is that in the last 10, 20, 30 years, dog transportation has become cheap. And people move their dogs in and out. And until a few years ago, I don't know exactly now, probably you know better than me, there was no mandatory treatment for Echinococcus multilocularis because it was endemic in both continents. So I personally moved my dog without that treatment. So in conclusion, what we have now is that the parasite is prevalent in wild hosts, and just so you know, this prevalence is very heterogeneous. There is one park that is called Beaumont in the city of Calgary, in which we have 9 out of 10 coyotes that are positive. And the 2 dogs that were found positive in the city of Calgary, guess what? They were both walked in the Beaumont. The European-like strain is prevalent, and the European-like strain is known to be pathogenic. And so the question is, possibly, my answer to the main question, is there a concern? We don't know yet, but there are some indications that may recommend to reassess the risk of human echinococcosis, alveolar echinococcosis. Thanks and sorry for...
Thank you. We have time for 1 or 2 questions or comments. Melissa.
Thank you for a very nice talk. Have you had any samples coming from British Columbia? And can you tell us about what you know from BC?
So we got a few coyotes sent from the Kate, help me, which is the office? That one. And we conducted, we screened some of them, I think about 10 of them in total, and we found 2 positive cases, and both of them were European-like. So these blue ones are. And these are multiple worms from 2 different coyotes. And the interesting thing is that when you take multiple worms from Saskatchewan, for example, from a coyote of Saskatchewan, you find the same genotype and the same haplotypes. But when you take multiple worms from the coyotes from BC, you find multiple haplotypes, you see, different haplotypes in the same coyote. So the impression is that the Echinococcus multilocularis, even if it is European-like in BC, it has been here for a longer time. And there is an hypothesis that was formulated by Jenkins and colleagues that this might be due to import of foxes for fur trade in the 70s or in the 60s. So that's why we see higher variability. But in both cases, we see European-like strains.
Any other? Yes, Chelsea. For the people online.
I was interested that you've hardly found any North American strain, because it seems like if this was an invasion, you might see an admixture of European and North American. Is it just that, is the North American strain confined to the far north, or has it been outcompeted? Like, what's your explanation for the fact that that strain seems to have kind of dropped off the face of the earth for your study?
So there are probably 2 or 3 reasons. One is that we are looking only mostly into coyotes, and a few cases of North America that we found were in foxes. So we are working on an hypothesis of host partitioning, so that the North American strain, sorry, the European-like strain is a new pathogen for the coyotes, but it's not a new pathogen for the foxes. So the foxes, even though they are competent hosts, they might fight back the infection a little bit better. And so in a naive host like the coyote, the European strain might strive. This is one hypothesis. We cannot exclude co-infection simply because every time you open an intestinal tract of one of these animals, you find hundreds, thousands, tens of thousands, if not hundreds of thousands of worms. And you cannot genotype them all. So we pick usually 1, 2, up to 10, no more than that. So if you have one of the 2 strains that is less prevalent in that specific host, then you wouldn't find it. What we are working on is with the droplet digital PCR and with deep sequencing to try to find a way to detect so to mix all the intestinal content, extract DNA, and see what is the proportion of the DNA of the 2 strains. But it's still a work in progress. So I don't know. I don't have the answer for this. But this is host partitioning is our working hypothesis, and we are collecting now animals, coyotes, and foxes from all over Alberta, thanks to work with the Alberta Trapper Association and the Alberta Conservation Association. So we are collecting data from across the province, and next year maybe I will be able to tell you more about this.
Thank you so much, Dr. Massolo, for raising our awareness about